Hi, my name is Manuel Corpas, and I'm really glad to be able to present today for this Kingston event. I'm afraid I'm unable to make it, but I promised my wife I would be in a holiday in Wales this week. So I'm afraid I cannot make it, but I hope I can make it next time. So I'm going to be talking about bridging the genomics greatest gap, which is, in my opinion, the diversity challenge. But before we go into that, I'd like to propose this particular story. So imagine this. We have a young mother in a remote uh, Ethiopian village. She rushes to the nearest clinic and carrying her infant who is unresponsive. The child was recently prescribed codeine for post-surgical pain management, but what was meant to relieve turned tragic. Unbeknownst to the healthcare providers, the child carried a genetic variant common in Ethiopian populations that make codeine, which is a very common drug, metabolize too quickly. Therefore, it was turned into a toxic substance. This story, unfortunately, is not isolated, and it underscores the pervasive issue in genomics, which is that there is underrepresentation of diverse populations in genomic research. While European ancestry individuals dominate datasets, the rest of the world, rich in genetic diversity, remains largely unseen. And this imbalance doesn't just skew scientific findings, it perpetuates inequalities in healthcare, leaving millions like that young mother and her child at risk of preventable harm. So today we face a challenge as well as an opportunity to bridge this gap and ensure that genomics serves everyone, everywhere. So this presentation is about unpacking the barriers, highlighting the urgency, and most importantly, exploring solutions to transform this narrative. And I want to start with this seminal publication that was developed by Alice Popejoy and Fullerton as her co-author, where they basically showed in 2009 and in 2016 the amount of genome-wide association study individuals, their ancestries. And what you can see is that from 2009 to 2016, in 2016, 81% of all of the individuals used for genome-wide association studies were of European ancestry. And you would have expected that this imbalance would have improved. Unfortunately, this diversifying progress has been slow. And as of June 2021, still, the proportion of Europeans had actually increased from 81% to 86% in 2021. So in other words, the proportion of samples from the represented populations have either stagnated or decreased. And you can see here in this graph, the amount of data from individuals, the number of individuals that have been used in GWAS, and the actual share of the global population. You can see here. So as you can see, there's an exponential growth of European data versus the other regions of the world. And if you actually look at the GWAS diversity monitor, last time I looked at it, you can see the countries that are represented. And in some respects, this diversity monitor, which monitors the number of individuals in genome-wide association studies, it shows in this particular vision that 94%, almost 95% of the total GWAS participants diversity is European. And I also want to draw your attention not just at these global imbalances of the different continents, but also I want to bring the attention to the fact that these labels that we use, European, Asian, African, Afro-Caribbean, Hispanic, or other, are tremendously inadequate to actually represent the diversity of these regions. And in a paper that has just been accepted in Nature Genetics, we showed that if we actually compare the amount of diversity that exists in the most representative and diverse reference set of individuals, which is the Simon's Diversity Genome Project, which contains 300 whole genomes from 146 communities and populations from around the world, especially with emphasis on representation of populations which are not well represented. If we actually take those populations and classify them or group them according to the Genome-Wide Association studies labels that we see in the GWAS diversity monitor, we can actually have an estimate of the diversity of the samples. In other words, we can actually calculate how diverse samples from this reference data set is actually represented in terms of diversity. So if we compare the amount of diversity contained in samples of Simon's diversity projects that are of European origin, you find that although the European global population is 9.4% of the global share, 19.1 is actually of the diversity is contained in samples of European descent from the Simon's diversity projects. If we look at all of the Asian populations of the Simon's diversity projects and quantify its diversity, we get a diversity of 19.4%, whereas the total Asian population is of 45.1%. And we can see that African populations, they are 17% of the global population, and they contain 25% of the global diversity. And same here, you can see the Latin American and Hispanic, and then other populations, the rest of the world. So we had two massive issues with the current classification labels, and this is what we are trying to explain in this correspondence. First of all, is that the diversity doesn't really correlate to the global share of the population, even though in this particular graph you can see the global population share, but you can see that it doesn't make any sense. So there's no correlation between the amount of diversity existing in a continent and the global share of the population. Then the other thing is that, as you can see here, we get like almost 20% of the rest of the world who are actually classified as other or mixed. And it is precisely these other or mixed populations, the ones that need greatest representation, because you can find a lot of indigenous populations that are very small numbers, but they contribute disproportionately to the amount of diversity that they could provide for reference studies. So this is tremendously problematic, and in my view, is actually increasing the perpetuation of disparities in representation of data. Then the other thing I wanted to mention is that countries from Hispanic or Latin American ancestry, if we actually cluster the populations in a single sort of label, if we then look at all of the different countries, it's a mess, because you find that within the Central American, Caribbean, and South American, you have different proportions of indigenous populations. You have countries like Peru and Bolivia, whose proportion of indigenous population is around somewhere between 70 to 80%. And then you also have that all of those countries, they all have an European proportion of ancestry, as well as African proportion of ancestry. So it doesn't really make sense to group all of the peoples from Latin America as Latin American, since they are actually a mixture of European, African, and indigenous American. So the diversifying progress, as I said earlier, has been slow. And the proportion of samples from underrepresented populations has actually stagnated. So why do we really need to have diverse genomic data sets? Well, because this diverse representation is essential for developing an accurate genomic databases as reference panels. So there are different genetic variants that are going to have different presence in different populations, and they may have an implication in our ability to diagnose and treat patients. And I'm going to give concrete examples of that. Having diverse populations in reference data sets are also important because they allow the better accuracy and applicability of genomic information for more precise diagnoses, treatments, and risk assessments. However, the representation does not only occur in genome-wide association studies, it also occurs in pharmacogenomics. So we performed an analysis of PharmGKB, which is the global repository of gene-drug reactions. In other words, it annotates and provides the list of all pharmacogenomic variants found that have an effect in our ability to metabolize drugs. And so the way PharmGKB classifies the rest of the world, note that the differences are significant with respect to the different labels on how genome-wide association studies classify continental populations. So for instance, here we have now subdivisions within Asia where you have East Asian, Central Asian, and Middle East, Sub-Saharan Africa. So we have very different boundaries. So if we actually quantify the number of individuals for which there is information in PharmGKB in terms of their ancestry according to these labels, again, we can see a tremendous bias. Again, we expect Europeans who are around 62% of the global share of data from pharmacogenomics studies, and the rest of the world incredibly underrepresented. If we were actually going to measure the percentage of under or overrepresentation, we also see that East Asians, compared to the global share of the population, are slightly overrepresented. And the least well represented in comparison with the global share of the total population would be South Asians, because there are 2 billion people from South Asian ancestry, and there's around 2% of the global share of gene-drug reactions individuals for which we have information for pharmacogenomics. Even though, for instance, we have the Oceanians, which are like 60 million people from around the world, there's a lot less data. But since the global share is smaller, then potentially they're less well represented. But as I was saying, the proportional representation is not a measure of diversity in genomic data sets. We also have here another example of clinical trials reported by the FDA, and you can see that the percentage of clinical trial participants by ancestry between 2015 and 2019, which is the most recent data that was available, 76% of all clinical trials were tested in people of European ancestry, 11% Asian, 77% Asian or African or African American, and then other. And again, you can see different ways in which the populations are actually tagged or labeled, which means that it is very difficult to have a consistent understanding of the level of underrepresentation that exists across different reference data sets, because they all have different ways of labeling. So the interoperability of analysis is quite challenging for this reason. And the genetic variability in genes encoding drug metabolizing enzymes contribute to the high number of adverse drug reactions observed in Africa. So let me give you this example, which is CYP2D6. So this is the most important pharmacogene. It metabolizes 25% of all prescribed drugs. And as you can see, if we then analyze the proportion of ancestry individuals and our understanding of whether they're able to metabolize quickly or slowly drugs that interact with this gene, we have a substantial amount of indeterminate individuals of African ancestry for which we have no idea of how any of these drugs are going to react according to their interactions with this gene. And this disproportionate amount of indeterminate activity is simply because there is more variation in the African continent than the rest of the world combined, and therefore our ability to understand all of those variants that influence metabolization of drugs for this gene is currently unknown. And therefore, people from African ancestry disproportionately suffer the consequences of potential higher metabolic drug reactions, which are adverse. So some examples, for instance, as I said earlier in my opening example, coding has been banned in Ethiopia because 30% of all Ethiopians have a duplication in this gene that causes severe adverse reactions. And therefore coding, since genotyping is not feasible in Ethiopia, this very common drug has been prohibited in this country. But there are also many other types of examples. For instance, there are 3 alleles associated with poor treatment for patients with breast cancer who are treated with tamoxifen, which is the most prescribed drug in some African populations. Another example is warfarin, which is the leading drug for cardiovascular disease and is basically an anticoagulant. And we find that is among the top 4 drugs leading to hospitalization from adverse drug reactions in South Africa and sub-Saharan Africa. There are, however, risk predictions that can be applied through algorithms, but they only work in European. And while it reduces the inaccurate dosing by around 18 to 24% in Europeans, for non-Europeans, this algorithm doesn't work. So again, we have another example where the precision medicine approaches only benefit a percentage of the global population. If we compare polygenic risk scores, which are a model for predicting genetic risk in complex diseases, we find that across 17 different traits from that have been used as a benchmark, we find that the further away in terms of genetic distance that you are from Europeans, the less precise these models are going to perform in terms of accurate risk prediction for complex diseases. What this means is that our ability to stratify high-risk individuals in underrepresented populations is compromised. And so, as a consequence, even many research studies do not include underrepresented populations because they are disregarded since they do not provide sufficient statistical power. And in order to dilute statistical inferences, there has been a lot of practice in terms of basically just focusing on Europeans that are going to give you the appropriate number of samples to be able to get significant results. So these biases result in associated medical practices that are being informed and benefiting only a subset of individuals. So in conclusion, I would say that it's absolutely imperative that the field's insights and benefits are available to everyone, irrespective of their ancestry or sex. I haven't given you some of the studies and results that I have for sex-specific issues in terms of diversity, because I didn't have time, but there are well-known documented issues for in terms of males being more represented, and as a consequence having providing disparities for women treatments. Moreover, it is absolutely essential to quantify the global genomics data underrepresentation biases for the development of precision medicine, because failure to do so can lead to inequities in healthcare, as well as lack of understanding of disease mechanisms in underrepresented populations. And I want to close with this quote from the International Covenant on Economic, Social, and Cultural Rights, which was approved by and accepted by the United Nations in the 60s, whereby everyone, irrespective of country, socioeconomic status, or whatever condition, everybody should benefit from the advancements of science. And with that, I want to say thank you. Thank you, Farzana, for inviting me, and I look forward to meeting you in person very soon.